Good afternoon, everyone. We are here for another week of biology, and this week we are going to be working on lecture five, which is on the topic of metabolism. Okay, so it has been a while since we had a live class. Well, I guess 1 week. Yeah, last week you were working on the lab, so we didn't meet, but we are back again. And this is going to be the last lecture before you are going to be writing the test number one. So today you have quiz two, which is 10%. You have until the end of the day to finish it. And for the test, that's going to be happening next Wednesday. So just to give you a bit of a heads up of what's going on, today and Thursday, we will do lecture five. Next Tuesday, we will do a test review. And then next Thursday, no class, just the test. Okay. And then you will have your reading week. Right? And then you're done for the first half of the course. When we come back from the reading week, we will be starting the second half. And just to remind you, once you finish the first test, you're done with all the materials. The second half is independent, it's on its own. You're not going to be evaluated on the material in the first half anymore. Okay, so there's that to look forward to. Okay. Fahima, do you have a question? I saw you unmute.
Test will be on class time only or the
No,
whole
it's a
day?
whole day. Yeah, six
Okay,
to yeah. So like, but then no class, so you can either use the time to do the test or you can use the time to study a little bit more. Okay.
okay, thank you.
Yeah. All right. So lecture five is quite complicated. All right. There are a lot of information going on, and I think just based on past experience, it's better if I teach this to you through writing notes instead of going through the PowerPoint slides. Okay, so we're gonna be writing a lot today and Thursdays. Please, take out papers and have some colors with you. Okay, we're not gonna be like drawing much, but there are information where I will use different colors to code them, so that it stands out a little bit for you. Okay, so I'll give you a moment to get that ready, and then we're gonna go. Okay. All right. So metabolism are basically the reactions that are going on inside a cell. And in lecture four, we learn about catabolism and anabolism. In catabolism, we break down molecules, and it's going to be harnessing energy from these molecules, and we generate ATP in the process. Whereas in anabolism, we're going to be building molecules that we need in the body. So even though there are many different kinds of reactions, what we are focusing on in lecture five is cellular respiration. Cellular respiration. Okay, so this is the main reaction for producing ATP in a cell. Now, we need ATP for various cellular work. Okay? Needed for cellular work, such as uh, mechanical work, transport, and chemical. Okay, so these are, just review from before. When a cell moves certain parts, like a flagellum or cilia, or if you think about muscle cells when they contract to create motion, those are mechanical work that requires ATP. Transport, we learned that in lecture 4. We know in active transport, when we move things from low concentration to high concentration, that's going to require ATP. And finally, chemical work, right? A lot of chemical reactions need supply of energy before they can happen. So all those things need ATP, right? Basically, without ATP, we won't have energy for the cells to work, and we basically would die from as a result of that, right? So we need to continuously make ATP, even when you're sleeping, right? This is like a non-stop process. So part of the reaction happens in the cytoplasm, and part of it happens in the now, can anybody tell me what organelles is responsible for carrying out cellular respiration? You should know that. You have quiz today, right? So
The
which organelle?
mitochondria.
Thank you, Cheyenne. That's correct. So part of it happens in the cytoplasm, and part of the reaction happens in the mitochondria. Okay, let me just move this kind of out of the way so it's not, you know, looking weird. All right, so I'm gonna write a word equation for you. Uh, I think you might have learned word equation in chemistry already. If not, it's basically you describe the reaction using words. Right? So in cellular respiration, we have glucose, and we're going to be reacting it with oxygen. And in the process, we will generate carbon dioxide plus water and energy in the form of ATP. Okay? I mean, I guess like I'm not gonna ask you for the full name of ATP, but like it might help to remember what it is. It's adenosine triphosphate. Okay? Adenosine triphosphate. Triphosphate means it has three phosphate in it, and you can break one off to release the energy if you want to use the uh, ATP molecule. Right? So that's the word equation. All right. Just give me a moment. I'll be right back. Talking too much increases my metabolism. Getting too warm in the hoodie. I need to take it off. Anyway, so that's a word equation, and now we have the chemical equation. Chemical equation. Chemical equation. Right? Which means we're gonna write the chemical formula. Does anybody remember the formula, chemical formula for glucose? We have been talking about it.
H6H12O6.
Excellent. Right? That is exactly it. Can you guys tell me like whether you've learned balancing equation yet in chemistry? Does this sound familiar? Balancing equation or not yet?
Not yet.
Not yet. Okay. So that's okay. I'm not going to ask you to balance it, but like I'm going to tell you what that means, right? So in any chemical reaction, the number of atoms in the reactant side has to equal to the number of reactants in the number of atoms in the product side. Okay, so like for example, if I have 6 carbons here, I need to have 6 carbons on the right because atoms cannot be created or destroyed in a chemical reaction. What you have on the left side has to be the same as what you have on the right side. Imagine buying a box of like a small box of Lego, and then you use all the pieces to build a car. If you take apart the car afterwards and use the same pieces to build a plane, right? It's the same building blocks, right? So you would expect to have the same number of red pieces in the car as you do in the plane. Right? So if I have 6 carbon here, I would expect to have 6 carbon on the right side. Okay, so similarly, there are 12 hydrogen here, but only 2 hydrogen here. So 6 times 2 gives me 12. And let me just quickly check the oxygen. 6 times 2 is 12 oxygen. Don't write this down. This is 6. So I need 18 in total. I already have 6 here, so I need 12 more. So you're going to learn how to balance equation properly in chemistry class, but the reason we do it is to obey the law of conservation of mass. That's what it's called. Okay, so that's the complete balanced chemical equation for the process of cellular respiration. And in the process, we're going to create 36 ATP. Okay, so ATP is not really part of the equation. It's just there to capture the energy that's being released from this reaction. Okay. So leave some space, maybe like 3 or 4 or 5 lines here, and we're gonna write a little bit more here first, and then I'm gonna add some stuff here. Okay, so leave some space. Cellular respiration is a type of redox reaction where electrons are transferred from one reactant to another. Okay, so that's what a redox reaction is. And there are different kinds of redox reaction, but they all have one thing in common. Whenever you have a redox reaction, there is a transaction of electron from one reactant, from one ingredient to another ingredient. Let's take a closer look at the word redox. Okay, so red or read, whatever you want to say, it stands for reduction. So reduction is gaining electron. Okay, we're going to be writing electrons like many times throughout the notes. So I'm just going to be a little bit lazy, and I'm going to put E with a negative because electrons are negatively charged, right? But that means electron. So reduction is gaining electrons. So that's what the red stands for. The OX, ox, stands for oxidation. So oxidation is losing electron. To remember this, this might help. Oil rig, like in the ocean, right? They have those big, huge platforms where they, drill down to the ocean and get the oil. That's called an oil rig, right? So you can remember oil rig. Oxidation is losing electrons, and reduction is gaining. Does anybody have any questions so far? It's a lot of information coming your way. So, if you are not sure about something, please be sure to ask so that you don't get lost for the rest of the class. Okay, well, let's go back to our reaction here. So it turns out, the reactants or the ingredients are glucose and oxygen, which means one of them must be losing electron while the other one is going to be gaining it, right? So the glucose is the one that will be oxidized. Okay, so it's going to be oxidized. It's undergoing oxidation, basically. So it's oxidized into carbon dioxide. So losing electrons. Now, those electrons have to go somewhere. Okay, we don't just let electrons fly around on their own. So ultimately, they are going to end up on oxygen. And when oxygen picks up the electron, it gets reduced. That's gaining electron. When it does gain the electron, it becomes water. And this process generates energy. And we want to store those energy until we need them. And we will store them in ATP. For every glucose that we react, under ideal condition, we can make up to 36 ATP. Okay? So essentially, electrons are removed, not just one, like a lot of them are removed. And ultimately, the electron is going to be transferred onto the oxygen. So that's the overview of cellular respiration with the equation and the oxidation and the reduction. Okay, I'm going to pause and let you catch up. And I know I ask you this like every 5 minutes, but do you have any questions?
Where do you get 36 ATP?
Where,
36. Why
where,
just 36?
yeah, why is it 36? Well, excellent question. I can't tell you right now, but we will later, okay? Because it takes like, an hour to explain, but you will understand by the end of Thursday. Okay, good
Okay.
question. Yeah. Okay, great. And I want to thank, the 11 of you who are, I recognize the names, right? You guys are the regular attendees of the class, so that's very important that you actually do come to the class, and I just want to thank you for being here. All right. So next part of the note, we are going to be saying we're going to be looking at four stages of cellular respiration. Okay. Now, let me try to explain this to you. I know we call this a redox reaction, but this is actually, you can also think of it as a combustion reaction. Okay. If you haven't learned that in chem yet, you will. Combustion reaction basically means you burn something, a fuel in the presence of oxygen, and then it produces carbon dioxide and water. So there are a lot of everyday situations where we burn things, right? For example, if you drive a car, the engine burns octane. That's the fuel that you put in cars. Okay? And I want you to think of the same equation, but instead of glucose, we have octane plus oxygen, and then you're going to burn it, produce carbon dioxide and water, right? So how an engine works is you burn the fuel a little bit at a time, and the explosion that you get from the reaction, it's what's causing the piston to turn, and then in turns the wheel turns, and then your car is going to move forward, right? So if you have a full tank of gas, you can drive, I don't know, 400, 500 kilometers, depending on how big your tank is. Okay. Now, same tank of gasoline, but if you imagine, um, like in movies, if a car blows up the engine, then you get like blows up like not the engine, the gas tank, then you get a really big explosion. All the energy in the fuel, instead of being released a little bit at a time, is released instantaneously, right? All at the same time. So that creates a big explosion, but it's not really productive work. Okay, like you can't get the car very far with a single explosion. And that's kind of what happens here. If we burn the glucose at one go, all right, in one big explosion, we're not going to be able to harness the energy from glucose in a meaningful way. Okay, you release the same amount of energy pretty much, but you do it at one instance. It's probably going to overheat the cell and kill it. So instead of having that big explosion, we are doing the same chemical reaction, but in four different stages. Okay, we're going to do it slowly, a little bit at a time. In fact, each of these stages will have many steps in them. Right? For the first stage, for example, has 10 steps, right? And then the second stage has like 3 steps and so on and so forth, right? So it's like a control way to extract the energy from glucose a little bit at a time. That's the key thing to understand here. All right. So I'm just gonna write down the names for you first. Okay, don't worry about the names at this moment. We will explain it later. Uh, I'm just gonna write it out. First one is glycolysis. Second one is pyruvate oxidation. Next one is Krebs cycle, also known as citric acid cycle. And then finally, we have the electron transport chain. Okay, I want to put a line here. I'll explain why later. Just copy down the names first. They are just names for the moment. Okay, we are of course going to explain the meaning of it and what's going on in each stage in more details um, as we progress. Now leave a bit of space here, okay? And then we're gonna go to the right side here, and I'm gonna draw a cell. This is cell, and this is the cytoplasm. Now, if you have been following along, then you remember the cellular respiration happens partly in the cytoplasm and partly in the mitochondria. All right, so I'm going to draw a mitochondria. Just, I mean, like obviously you have a lot in a single cell, but I'm just going to draw one, and this is not to scale. So there's mitochondria, and you might remember it looks like this on the inside, right? Like it's all folded up. That's the mitochondria. Okay. ION for singular, IA for plural. So over here, we have four stages. Glycolysis, that happens in the cytoplasm. Step 2 and step 3, they happen in here. Does anybody remember the name of this part of the mitochondria? Like the innermost portion of it?
Matrix.
That's right, that is the matrix. Thank you. So 2 and 3 happens there. And number 4 happens right there in the cristae, the foldings that you get from the inner membrane. Remember, I told you to increase surface area, the inner membrane is going to fold up, right? Like this. It's going to fold up like an accordion. So you can pack more stuff inside these finger-like projection, these foldings, they are called cristae. That's where you will find the electron transport chain. Um, Today we probably have time to talk about 1, 2, 3, and then we will finish up with number 4 on Thursday. Okay. All right. So what's going to happen in 1, 2, 3? I mean, lots of stuff's going to happen, but the theme is that, right? We are doing a redox reaction, okay? And in this reaction, we are oxidizing glucose, which means we are going to be ripping electrons from them, from glucose. Okay. Now, these are not ordinary electrons. These are electrons that are packed with energy. Okay. So in the first 3 stages, glycolysis, pyruvate oxidation, and Krebs cycle, high energy electrons are removed from glucose. These high energy electrons are captured by, they are captured by energy carriers, energy or electron carriers. You might hear people say both, okay? Uh, they mean the same thing, okay? Because these electrons are packed with energy. So if you carry the electron, you're essentially carrying the energy. So they are captured by energy or electron carriers and will be taken to the ETC. I forgot to mention, okay, the short form for electron transport chain, because it's such a long name, is just ETC. Will be taken to the ETC for ATP production. Okay. So basically, we have we have electrons that are being released from these steps, and we get the electrons from the glucose, and the carrier will transport them to ETC. Yeah, we'll take them to the ETC. Like you might have another one being released here, and then you're gonna take them to the ETC. So let's talk about the two important electron carrier, two types of electron carriers. Okay. So one of them is called NAD plus. Never mind what it stands for, because the name is really long. I'm never going to ask you for it. And then the other one is called FAD. Okay. There's no plus for this one. And what happens is the NAD plus can pick up electron, and once it picks up electrons, it's going to become NADH. And now you might wonder, like, where does the H come from, right? Uh, we're not going to go into all these details, okay? The H is just around. The important thing is once you put the E in into the NAD plus, then it becomes NADH, okay? And what happens is when it gets to the ETC, the electron can get off, and then it will become NAD plus again. Okay? So this is like without the electron, this is with the electron. Similarly, for the FAD, you can pick up electrons with it. And once you do that, it becomes FADH2, just the name. That's the full version, packed version. And it's going to take it to the ETC, and then the electron's going to come off. Okay. So this here, when it picks up the electron, it's gaining electron, right? So this is actually reduction, and then this over here is oxidation. Okay, same thing. Reduction and then oxidation. Now, to help you understand, I will draw this picture. You don't have to draw this next part, okay? Like if you want it, you can add it on later. But like just listen to me, uh, because I think it's more important than trying to draw this out and then miss what I say. So this is just an analogy to help you understand. Okay, so let's say we have like a hotel or something here. We'll call this hotel number one, and then we have some people waiting, and then here is like hotel number two. We'll also have some people here, and then we have hotel number three. Okay. So, think of this as like some kind of vacation destination, all right? So people go to vacation and they go to these different hotel, but now it's time to go home, so they have to go to the airport. Okay? They have to go to the airport, which is number four here. I'll draw an airplane here. Okay? So imagine, like people on vacation, how do they get from the hotel to the airport? Like obviously you don't drive your own car, right? So what do? You call a taxi, right? Okay, you call Uber or something like that. Right? So the taxi that's gonna come and pick them up is basically the NAD plus. Okay? Don't draw this, just listen to me, okay? Um, to explain this. So the NAD plus is gonna pick up the passenger. In this case, the passengers are like the electrons. Okay, so these guys get on. All right. How do you know a taxi has people in it, like in real life? Um, like how do you tell? Like if a taxi is coming from far away and it's dark, you cannot see if there's anyone inside. Like how do you know if someone is in it or not? Do you guys know? Like I mean, I haven't been on a taxi here, but like I see it on movies, right? Yeah, go ahead, Cheyenne.
Don't they usually have like a light on the
They,
roof or
yeah,
something?
exactly, right? The lights, right? So the light's gonna come on and say occupied, right? When people are in it, right? So that's exactly what happens, okay? The NAD plus the car, oops, I erased the whole car. Let me try that again. Okay, so there is the NAD plus. Okay, the electrons, the passenger is gonna get on. Once they get on, it's now full, so it becomes NADH. Putting the H there, it's like turning on the light, telling people, hey, it's full now, okay? Don't get on anymore. And then where is this guy going to go? Well, it's going to drive these people to the electron transport chain. And then the passenger is going to hop off. The electron's going to hop off. Wrong color. They're going to hop off. And then the taxi is free again. So let's turn off the lights. Let's change back to NAD plus. Okay. And what's going to happen to this guy? Well, it can go back and pick up more people, right? Like maybe from number two, it doesn't have to. It can go to number three and pick up some, and go to number one and pick up some. And it's just like a shuttling surface that keeps on going back and forth, right? And transporting these electrons from one place to another. Right? It just goes back and forth, like switching between NADH when it's full, and then letting them off and becoming NAD plus again. Does that make sense to you guys? Like any questions? Same thing with the other guy, right? The other is the FAD. So FAD is empty, right? It can go and pick up passengers and it becomes FADH2, right? So essentially you have two shuttling surfaces here, the red car and the blue car. They take the electrons that are released from stage one, stage two, and stage three. They take them to the ETC, and then at the ETC, at the stage four, that's where you're going to harness the energy from the electron to make a ton of ATP. Okay? 36 to be precise. So that's that. Okay. Again, you don't need to draw this. It's just an analogy, hoping it helps you understand better. Okay. But two types of energy carrier. Okay. So how much energy do these carrier actually carry? Okay. So this is important. Important. I'll highlight it for you. Okay. I always highlight it when I write this, but then people always forget by the time I ask them about it later. Okay. So like maybe double highlight it. Okay. Double highlight it if you can. Okay. Very important. Each NADH contains enough energy from the electrons to make 3 ATP. Again, like I'm not going to go into detail of like how do you know, like how do you know it's 3 and not some other numbers? Well, people did experiments and they found out basically. That's the short answer. And each the other guy, which is FADH2, contains enough energy to make 2 ATP. Okay. So write those down. Just give me a moment. I was looking for the concept check question here. Oh, there it is. Let me just copy this guy over. All right. So, what do you think? Let me do the polls for you guys. If you don't see the poll, you can just type the answer in the chat box. All right, go, go, go. All right, that was like almost a minute. I got like about 5 responses. That's okay. I'm just gonna end the poll. So here we have 4 NADH. 4 NADH. Now it says each NADH contains enough energy to make 3 ATPs, right? So I multiplied this by 3. 3 ATP per NADH. 3 times 4 is, of course, 12. Right? And then we have 3 FADH2 multiplied by 2 ATP per NADH. Okay. Again, like why is this 3? Why is it 2? It's just like something that you have to memorize, right? Just kind of take it for granted. It's numbers that I give you. So 2 times 3 is 6. And when you add them together, 2 plus 6 is going to give you, sorry, 12 plus 6 rather, it's going to give you 18 ATP. So that's the answer. Does that make sense? Okay. I mean, only 5 people did it, and only some of you get it right. But for the rest of you who didn't do it or didn't get it right, I hope this makes sense, okay? Because you're gonna have to do some of these calculations on the test next week. Okay, so that is that. Let's do the next note, next part of the note now. Next part of the note. All right. 2 ways to produce ATP in cellular respiration. 2 ways to produce ATP in cellular respiration. So method number 1, just going to use Roman numerals here to separate it from the 1, 2, 3 that I did before, okay? Doesn't have any special meaning. So this one, it has a long name. I am very sorry. Okay, I didn't make up the name. If I get to name it, I wouldn't call it this. It's really long. Okay, so this is called substrate level phosphorylation. That's how you pronounce this word, okay? Substrate level phosphorylation. Just on a side note here, to
SUP,
yeah,
can you spell this again?
sorry, yeah, I know my writing, right? Substrate. Okay, substrate is like the thing that you fit in an enzyme, right? Do you remember? Okay,
Ingredient.
yeah, the ingredient, exactly. Very good. There you go. So substrate level phosphorylation. Look at what does that word even mean, right? So to phosphorylate something, anything. You can phosphorylate a molecule, you can phosphorylate a protein. Okay. To phosphorylate something means to add a phosphate to it. To add a phosphate group. Okay. Phosphate, again, I'm hoping you learned this in chemistry, is PO4 negative 3 to it. Okay. So to phosphorylate a protein means to add a phosphate to it. To phosphorylate, okay, listen to this part. I know you're busy copying, but To phosphorylate ADP means to add a P to ADP. And what do you get afterwards? Let me say that one more time, okay? Listen. To phosphorylate ADP means to add a P to it, to add a phosphate to it, okay? What do you get in the end?
ATP?
Yeah, because ADP is diphosphate. We learned about this in lecture 4, believe it or not. And when you add the P on it, then now we have 3-phosphate and we get back our ATP, right? So substrate level phosphorylation is about adding a phosphate to ADP. And in the process, we're going to generate ATP. And this is how it's done. Okay? In substrate level phosphorylation, it is a direct production of ATP where an enzyme transfer a phosphate from a donor, or sometimes it's called like a fuel molecule, onto ADP. Just copy it down first. I'll explain. Okay, so basically, this is what it looks like if I have to draw it out. There is an enzyme here. Right? We know enzyme. You should know enzymes are proteins that helps catalyze chemical reaction. They help speed up chemical reaction in the body, right? So we have a fuel molecule here. Okay, this is the fuel molecule or donor molecule. Doesn't matter what you want to call it. Okay, it's fuel or donor molecule. There's the enzyme. So the fuel molecule has an extra phosphate. Well, not an extra phosphate. Like it has a phosphate that it doesn't mind giving away. And we have ADP right here. ADP. It's basically used up ATP. It's like a drain battery. I told you that before. And it needs to be recharged. Right? So what the enzyme does is take the P from the fuel and it will add it back onto the ADP. And at the end, you're gonna get ATP. That's that, right? Make sense? This accounts for about 11% of ATP produced in cellular respiration. Okay. How many ATP are we supposed to make from one glucose? What's the magic number? Does anybody remember? 36, that's correct. Okay, so this accounts for about 11%, which is like 4 of them, okay? So not a common way, but it's like a direct way of making ATP. Okay, so because this is like so long and it's difficult to spell and write, I'm just gonna put SLP from now on, okay? You don't necessarily have to memorize the name, I guess, but when I say SLP, you should know, oh, this is the uncommon way of making ATP. Okay, we're just gonna be using it a little bit, not all the time, just making 4 out of the 36 ATP. All right. So we're talking about 2 different ways to produce ATP, huh? This is one of the ways. Method number two. Number two. It is called oxidative phosphorylation. Okay, OP if you would. Oh, by the way, I'm so sorry. I forgot to write another point here. Sorry, let's just go back to SLP for a second. This accounts for about 11% of ATP produced in cellular respiration. Only happens in 1 and 3. And remember, we have 4 stages, right? If you don't remember the name, that's okay. We're gonna write them out later. But it's gonna the substrate level phosphorylation is only gonna happen in the glycolysis and Krebs cycle. As for the oxidative phosphorylation, okay, or OP, this takes like at least half an hour to explain, and we're not there yet, so we'll explain it on Thursday. But this accounts for about 90% of ATP production, of ATP produced in cellular respiration. Okay, I know, 90 plus 11 is not, it's like over 100, but 90 is easier to remember than like 89, right? It's like approximately anyways. So like don't get too hung up on that. This only happens in ETC, step 4. Okay, the airport, if you would. So it's kind of like an indirect way of ATP production. Okay, you're not making it like immediately. It takes some steps in between before you can actually get ATP. Okay? Whereas in the substrate level phosphorylation, it's just one single step. It's very simple. You take the P from the fuel and you add onto ADP, you're done. But for oxidative phosphorylation, you're gonna go through a bunch of steps in between before you can make ATP. So it's like indirect. Okay? So that's that. Yeah, so that's kind of like all the background information so far. Anything you want to ask me at all before we move on? No? Okay, great. I think we're gonna take an early break today. It's already a lot of information as you can see. We will resume in 7 minutes maybe. Okay. So when we come back, we will continue this note, but we will also be doing an other note parallel to it. Okay, so you need like 2 pieces of paper side by side. All right. See you in a bit. Right, let's keep going. Now that we have a background understanding of what the cellular respiration is about, we are going to talk about what exactly happens in each of the 4 stages. Yeah, like I said, we will have time for the first 3 stages today, and we'll do the 4th stage on Thursday. So this is stage number 1, which is glycolysis. So glycolysis happens in the cytoplasm. Okay, like this is the cell, nucleus, and then all this stuff over here, that's the cytoplasm, right? So it happens in the cytoplasm, and it is anaerobic. Anaerobic, meaning does not require oxygen. So sometimes, like during strenuous exercise, for example. Imagine running at full speed. There's going to be a point where your leg muscles are using up more oxygen than you can deliver. So in those instances, glycolysis can still happen even without oxygen. Right? We'll talk more about those situations next class, but that's what anaerobic means. What else? Okay, so for glycolysis, the word glyco means glucose, and lysis means, do you guys remember what lysis means? We learned this like a couple times already. Just, want to see.
Like death or destruction?
Uh, yeah, I think you're thinking about like the cell popping, right? Like dying. In that context, is that correct?
Yeah.
Yeah. Okay, so it's close. Okay, lysis means to break. Okay, so we learn lysis in like hydrolysis to break something with addition of water, or when a cell swells up and break, we call it lysis. So glycolysis is to break glucose, basically. So a glucose, which has 6 carbon in it, is going to basically break down into 2 molecules of what we call pyruvate, okay, which is 3 carbon each. Sometimes you might see notes or textbook, they say pyruvic acid. For us, it means the same thing. Okay, so I'm just gonna write pyruvate here. 6 carbon become 2 pyruvate, 3 carbons each. All right. So the glycolysis actually has 10 steps in it. Let me see if I can show you a picture of it. Give me a moment. Oops. I don't know what I pressed. Too many things open here. Just give me a moment. There we go. Okay. Of course, all these things are like in the PowerPoint slides, right? You're encouraged to go through them yourself. I just think it sticks better if I write it all for you. So this is the actual glycolysis with 10 steps. Okay, and the names are really complicated. You have glucose becoming glucose 6-phosphate, and there's an enzyme that catalyzes it. So it's very complicated. So of course we're not gonna go into the details of all of those things, but the important thing to know here is that in glycolysis is separated into 2 main phases, if you would. Okay, so we have the energy investment phase. Okay, that's pretty much like step 1 to 5. And then we have the energy harvest phase, which is step 6 to 10, basically. Before we learned that you might remember this graph, that you need to supply a bit of energy before a reaction can occur, right? This energy is called like activation energy, right? We need to basically energize the ingredients before they can react, right? So that's what the energy investment phase is about. Okay, 2 ATP molecules are used to energize glucose so that it is going to react. You might have heard the phrase, it takes money to make money, right? Basically, it means that, if you want to make money, you got to invest some money first, right? Like you use the money, you put the money in a savings account, then the bank's going to pay you interest, right? So you put in $1,000, maybe the bank's going to pay you like $10 or something, right? Of interest. So you have to invest first, and then you get some money back. So in glycolysis, we invested 2 ATPs to energize the glucose, and in the end, in the energy harvesting phase, we're gonna get 4 ATPs are produced directly by substrate level phosphorylation. That's the method where there is an enzyme and it transfers the phosphate from a donor to ADP, right? So 4 ATPs are produced, and also high energy electrons are released from glucose and captured by 2-NADH. So that's actually a pretty good investment. If you invest 2 ATPs and you get 4 ATPs back, how many ATPs did you actually make? It's not a trick question. Just want to make sure that you understand this. If you invested 2 ATPs and you get 4 back, how many did you actually make? 2, exactly, right? So we say even if you produce 4, but that is actually 2 net ATP. 4 is called gross. Gross is before you subtract your expense, right? Like your gross income. It's like before tax and everything, right? So 2 net ATP. And we have NADH, which we know is going to be used to produce ATP, right? Can someone tell me where these things will go? Where will they go? Go to where? Take a look at the notes. I assume you've been copying stuff down. The NADH is like the taxi, lets the electron get on. Where do we take them? Where do we take the electrons? Okay, let's go back and see, right? So high energy electrons are removed from glucose. These electrons are captured by electron or energy carriers like the NAD and the FAD and will be taken to the ETC for ATP production. Okay? So these NADHs that we produce here go to ETC for more ATP production later on. You with me, guys? All right. So I want you to please have another piece of paper. Let me show you what we're gonna do. Okay. Nope, that's not what we're gonna do. Yeah, we're gonna do this chart that summarizes everything that's happening in cellular respiration. Okay. So we won't be able to do it in one go. We're gonna add stuff to it as we continue. So I need you to have a separate piece of paper next to you. All right. So we started with glucose, and I want you to remember glucose has 6 carbon, C6H12O6, right? 6 carbons there. And what's going to happen is we are going to be investing some energy into glucose. It takes energy to make energy. How much energy are we investing? Well, take a look at the notes. We wrote down 2 ATP. Okay, so you're going to invest 2 ATPs, and of course, afterwards, they become ADP. They are drained, they're used, right? Okay, so that's like first 5 steps, which we just summarized into one single step here. So this is glycolysis. And this is the energy investment. Sorry, it's really hard to write sideways. Phase. And then it's gonna split at some point. And after it's split, a couple of things gonna be happening here. Each path will now produce 2 ATP. So, we're going to take the empty ADP. The enzyme's going to help us put phosphate onto them, taking it from a donor. Same thing's going to happen on this branch. All right. And at the same time, we're going to be releasing high energy electrons, and we will be capturing them with NAD+. Okay, so there you go. This here is it's also glycolysis. And this is the energy harvest phase. So you invested 2 ATPs, but you get all these things back. Okay, you immediately get 4 ATPs, and then you mix some NADH, which you can cash in later at the ETC, right? So the end result are 2 molecules of something called pyruvate. Pyruvate. We are splitting the glucose. Glycolysis. The lysis, the breaking of glucose. And how many carbons for each of the pyruvate here? Started with 6. Break it in half. You have 3. And this happens in the cytoplasm. Questions at all? Okay, that's that. Let's go back to our note then. And we'll add more to this as we go on. Okay, we're almost done. Okay, maybe 20 more minutes and then we're done. Let's go back to here. So that's stage 1, glycolysis. Stage 2. Which is pyruvate oxidation. What does the word oxidation mean again? Pyruvate oxidation. Are we adding electrons to pyruvate, or are we removing electrons from pyruvate if it's being oxidized? Removing, that's right. Because remember oil rig, right? Oxidation is losing. So pyruvate is going to be losing electrons, which is the theme here. The first 3 steps, I'm going to go back here again. The first 3 steps is all about removing electrons, capturing them, then transporting them to this to the ETC. So in pyruvate oxidation, what happens? So if O2 is available, the pyruvate molecules from glycolysis will move into the matrix. Remember, we drew a cell, and I told you glycolysis happens in the cytoplasm. 2 and 3 happens in the matrix. Okay, so if oxygen is available, that's where they're gonna go. They're gonna go into the matrix. Okay. Each pyruvate will lose a CO2 and produce one NADH. Okay, so remember, that's each pyruvate. But from a glucose, you split it into 2 pyruvate, right? So even though each pyruvate produces 1 carbon dioxide and 1 NADH, it's actually 2 carbon dioxide and 2 NADH per glucose. And that's an important distinction to remember. Okay, let me just quickly recap because it's important to know what has happened so far in order for the next step to make sense. Now we have been producing quite a bit of, energy carriers as well as ATP, right? But how much did we invest at the beginning? What do you think? Do you remember? How much did we actually invest in this whole process? How much energy have we put in so far? Like at the very first step, right? Like how much energy have we invested in the investment phase? Yeah, 2 watts though, specifically. 2 ATPs. Yeah. Okay. Are you guys lost? Like, I feel like I know it's a lot of information, but uh, are you guys okay? Like, I feel like I'm not getting through to you. If anything you want me to clarify before we move on, because it's only getting more and more complicated.
Still complicated.
But do you have a specific question about it though? Like, is there something that not just you specifically, Fahima, but like, in general, are there something you guys confused about? You want me to explain again? Or it's just too much information? Hmm?
Too much information at a time.
Yeah, so it takes time, right? I guess, yeah, it takes time to digest the material. All right, so let me help you put this into perspective again. We started with glucose, right? Okay. And then
Yes.
because we want to break it down, we want to harness all the energy from glucose to make 36 ATP, right? But you cannot get something without putting some effort in, right? Without putting some energy, some investment in, okay? So we've invested 2 ATPs at the beginning to energize the glucose to prepare it for the reaction. And from that investment, it's like you're paying $200. From that $200, you actually got back quite a bit, right? We got back 4 ATPs and we got some energy carriers like NADH, 2 of them from glycolysis. And remember, each NADH is equivalent to 3 ATPs, right? From the important note right here, right? Each NADH contains enough energy to make 3 ATPs. So, it's quite a good payback from the initial 2 ATPs. And then from the pyruvate oxidation, we are getting even more NADH from it. Right? So what I'm trying to get you to think about is we've only invested 2 ATPs at the beginning, but we have been getting quite a bit of return. So at this point, the investment we have put in at the beginning is kind of used up, if you would. Okay. the molecule is no longer energized like before, right? So it's not really willing to react further unless we invest more again, okay? And that's what the third step is going to do in pyruvate oxidation. A molecule called coenzyme. Coenzyme A is added to pyruvate to re-energize it. Okay. As I said, we've extracted enough stuff from it, and it's just not willing to go any further. You kind of have to recharge it. You need to refill, the tank, so to speak. All right, and that's all for pyruvate oxidation. So on the other note, let's see what happens. So the pyruvate, each of the pyruvate will have three reactions happening to it. Okay, first thing is we're going to remove a carbon dioxide. How many carbon dioxide are we supposed to produce in cellular respiration? Take a look at the equation you wrote down. How many carbon dioxide?
6.
6. Right. We balance the equation, right? So 6 carbon dioxide. So here are 2 of them. And at the same time, we are going to be removing high energy electrons from the pyruvate. And anytime you remove electrons, you need to capture them. So that's what the NAD plus is here to do. It's going to pick up those electrons. And where are we taking the electrons? To stage number 4, right? To the ETC. So we can make even more ATP. And by now, we've gotten enough returns. We have to invest more if we want to make more energy. Okay, so that's what the coenzyme A is about. Coenzyme A, it's like a recharging molecule. Coenzyme A. So after all these reactions, all 3 steps, you end up with another molecule called acetyl-CoA, right? So this summary chart is it's a good thing to have next to you when you're doing the test, right? Just as a reference, because I know it's really hard to memorize all these things, okay? And I don't expect you to, okay? Just having the reference sheet there is going to help you through, some of the questions. But do tell me this, how many carbons do we have in acetyl-CoA? And we started with 6 in glucose, we split it so it becomes 3. And then now, how many carbon atoms in acetyl-CoA? Look at what happened along the way. Tell me how many carbons are left.
Green.
Yeah, it was 3, and then we remove a carbon dioxide, right? So carbon dioxide has 1 carbon. So 3 minus 1 is 2 carbons, okay? 2 carbons. All right. You know what? I was going to do Crab Cycle as well, but I think it's probably a good place to stop, right? It's already a lot of information. You definitely need to, sit down and digest these things first. And you also have the quiz to write. So we're gonna end here for today. But on Thursday, we will finish the rest of lecture 5. And like I said, next Tuesday we'll do the test review to prepare for the test next Thursday. Okay. Thank you for coming again. Take care, guys, and good luck with the quiz. Bye for now.